Bon, on va passer au séminaire. Donc Matthieu Piel, qui est exactement à l'Institut Pierre-Gilles de Gênes et qui est membre de l'unité de biologie moléculaire de l'Institut Curie, qui est un peu la personne qui m'a poussé à m'intéresser au volume du noyau. C'était il y a très très longtemps. On n'a pas réussi à faire grand-chose il y a très longtemps. Puis c'est revenu il y a 3-4 ans et depuis, on collabore de façon très heureuse, enfin au moins pour moi. Voilà, donc je lui donne la parole. Merci Matthieu.
D'ailleurs, je vais... I will switch to English. Maybe you told me that maybe some people prefer to follow the seminar in English, so I switch to English. Yeah, so actually I will show one slide on this thing. We started to talk about volume was really when we looked at the volume of cells in mitosis. We still have not final answer on that. But what's quite amazing is with in particular with this student that Jean-François mentioned, Romain Rollin, who joined for his PhD for 3 or 4 years ago. Initially to understand the blebs of the nucleus, and he did not understand, and he took really time to go through the literature, and in the end what he understood basically explained everything we are doing in the lab for the last 10 years. So it's really quite amazing. So we are not done at all going through like all the consequences of these models that Jean-François exposed. But so what I will show you, I focused my talk mostly on the volume of cells. We have also a whole part on the volume of nucleus, but I'm not talking about it. And what I chose to do is to try to go through the different time scales which are involved in changes in volume of cells and mammalian cells. I'm just going through mammalian cells. So the beginning is really go fast because you add all these equations explained to you in details. So the volume of cells, it was kind of understood for the basics really already a long time ago. And really when you have these equations, it's actually, I don't know if you would say that in your previous course, but they are very general. You can imagine all kinds of things, but they become very simple when you have an idea of the numbers and the simplification that Jean-François mentioned, that basically if you have a cell, at least a cell in culture, again, that's if you are in a tissue, you can have the outside much more complicated because the concentration can change outside. And there are people who have been working on that here. Like Hervé Turier, he was here before, but I'm not sure if he stayed. But let's say for cells in culture, you can consider it's constant outside. And inside, you have mostly potassium. Chloride is very low. The charges on average are -1. And you have this amount of biological molecules, let's say, which, as Jean-François said, are mostly amino acids for the amount of it. So with all that and with the fact that you can assume that the osmotic equilibrium is achieved because there is very little tension on the membrane, you can write the volume like that with the excluded volume. And so here, let's say I will show you how this is really well experimentally well supported, this relation. Okay, and I think it's also a sort of the fact that this relation is so well supported is really a sort of experimental proof that the cell is operating in a sort of diluted regime where you can apply Van 't Hoff simple relation, because otherwise you would not have a linear relation like that with the volume. So why we started to work on cell volume already probably 15 years ago, because Maël Lebert had this idea, which was actually already out from other lab, to measure the volume of cells just using the microfluidic chambers we were using for all kinds of other experiments, and putting fluorescence in these chambers and integrating the loss of fluorescence over the cell to get their volume. So it was always a sort of side project in the lab, but you see that over the years it involved quite a lot of people, so we have accumulated a lot of things. And so this method turned out to be really nice because you can measure the volume of cells with just one plane of imaging with a low magnification objective. You can have many cells. It is not sensitive to the focus, and it's robust to, I mean, it doesn't depend on the shape of cells. And we really found that the relative volume precision that you can have when you measure the volume several times of the same cell is around 1% or even below that. Okay, so it's very precise. Okay? And so with that, we did the basic experiments that you can do when you want to modify the volume of cells, which are the osmotic shocks, which have been done for a long time, but usually not on single cells, not with this precision that you can get. And here you can really look at if you put, you change the medium outside, that's how the cell here is shrinking or swelling. And you can have the imaging on many cells with a time resolution which is below, I mean, on the more like tens of millisecond scale. You don't really need that here, but you see that the volume change happens in the second range. Okay, so that's the time scale of water getting in or out of the cell. And then you have a longer term change of volume, which is also well known for decades, which is that after the cell has swollen or shrunk, for the next tens of minutes, they adapt their volume, so they go back to their initial volume, not perfectly, okay? So the adaptation is not perfect. I think this is not very well understood. And also here you can see that the cells we are looking at, there is a third type, so there is a water flux time scale, this is the ion fluxes time scale, okay, tens of minutes, and then you have the growth. You see that all the curves are going up here because on longer time scale the cells are growing. Okay? So this is basically three obvious time scales you have when you do this kind of experiments. And so here is just some quantification of the sort of fluxes that you have. So for the passive volume change, it's a few micrometer, 100 of meter cube per second. The adaptation, which is active ion pumping, is 100 meter cube per minute. Okay, and you can measure that as a function of the difference of osmotic pressure that you apply. And here there is also one thing you can do, which is a question that often people have. We just did it, but we did not really study it. Is how much membrane there is. So when the cells swell, for example, is it really that you will tense the membrane as it swells, or is there a limited amount of surface area of the plasma membrane? And what you realize when you do a shock like that, if you stain the membrane, you can see that the staining here becomes very dim and regular, which means that a lot of membrane folds have opened. And if you measure, you can use this volume measurement combined with a fluorescent molecule that will become fluorescent when it gets inside the cell, but it doesn't get inside the cell, it cannot get through the membrane. So it will get inside the cell only when the membrane opens. And so you have exactly at which moment the membrane opens, and you can see that actually it's consistent with the moment where you measure with the excluded fluorescence, you measure a drop in volume, because of course the fluorescence that you have put outside is also getting inside. So here you can have your maximal volume swelling before the cell explodes, basically opens the membrane, and this is usually 5 to 6 times the initial volume. So the surface is about 3 times the initial surface area. You have a lot of available surface on your cell. Okay, and this is within seconds, so the cell really doesn't have time to put more membrane to accommodate. That's what the Actually, I always thought it would be interesting to also test that on different cells, because I don't know if this is always the case, or some cells maybe have more or less, I don't know. But it's a very easy experiment that would tell you how much membrane you have readily available to swell or to shrink.
How is it stuck, this membrane?
It's folds. Little, you have some sort of pili, you have lots of pili, depend on cell type, but also a lot of folds. And there is actually it's been seen already, and you can estimate that because the membrane is anchored on the actin cytoskeleton with a certain distance between the points, and between each point you have a sort of little fold of membrane like that, and so it would give you about 3 times. I think this is very this is very small.
This is very small.
Amount, yeah. This is like 1%, or a few percent, yeah, I think. So here, with this, if you look at these volumes that the cell have at the max or min when you do an osmotic shock, you can draw a plot it's called a Ponder plot, because I think Ponder was the first to do it. And it just basically is this verification that I told you about of the Pump-Leak model, that you have a linear relationship. So on average, the cells are perfect osmometers, and you can see that if with this drug you are removing the actin cortex, so it doesn't change anything. And with this drug you are killing one particular type of pump that is important after to adapt the volume, and here it does nothing. Okay, so it's a passive response, doesn't depend on the cortex or the pumps, and it's very linear. And the slope is 70%, which means that the excluded volume is 30%. So it's more than what you were saying. I don't know what you said, it's very small. Let's say it's not so small, actually. So you could imagine that if you are doubling the amount of things you have inside, you will start to get to a limit that you would be too crowded. And that's why I think you also have, as these cells are operating in a regime where proteins can be easily crowded, as you heard last week, I think, from Morgan Delarue. Because actually there is 30% of the volume. And this 30%, I'm not doing that here, but you can estimate that it's really not the volume of the sort of backbone of the protein, is really the volume of hydrated protein. If you do a little calculation, it gives you a 5 nanometer diameter for proteins if they fall on each other, and they represent 30% of that volume. And it's about 0.3 kilogram per liter. So that's, I think, what is nice when you have this is to show a counterexample, which is a yeast cell which has a very rigid cell wall. And if you do the same experiment with a yeast cell, you will get something that's really not linear at all. Because here what really dominates is the stiffness of the cell wall. And it's enough to remove this cell wall to make what's called a spheroplast to recover a linear relation. Okay? And that's a little bit what you get for the cell nucleus, actually. It's not non-linear relation because of the nuclear lamina. So you can ask then, in different contexts, how much the cells change their volume. Okay? So here I showed you the osmotic shock, which gives you a bit the time scales at which things can change. So one thing, of course, that we do a lot in the lab is looking at cells that move. And you can put cells in the same sort of volume exclusion chamber, but you fill the chamber also with a collagen gel. And collagen occupies a very small fraction of the volume, so you can still apply exactly the same technique. And you can follow the volume of the single cells as they move. So each one of these is a single cell, you see, that moves in the collagen gel. And when you zoom, you see this is a cell that is extending some protrusion and moving around. So here you have the time of the cell, and here you have its volume. And you see that most of the time is really constant. And then you have sudden kind of drops up and down, so on. And that usually corresponds to times when the cell is extending protrusion. And so here, if you look at cells that move more or less fast, if the cells are really moving very slowly, basically they have a completely constant volume. And the fast-moving cells, they can fluctuate their volume plus minus 10%, let's say. And you have this sort of correlation that's not so nice, but between the speed and the... Okay, that tells you that the volume...
Naive question. Why is it that we don't see the increase that you showed before?
What do you mean increase?
That you showed us that the cell volume increases as a function of time.
Oh, yeah. So here first, you see... Yeah. So this is a very good point. Really, I'm proud of you. You're almost a biologist. So actually, these cells are differentiated. These are dendritic cells, so they don't grow. Okay. They don't grow. Good point. It's a good. I never thought of looking at that there, but this over this time, if we were looking at proliferating cells, we would see the growth. It's true. Yeah, because the growth is about 5% per hour in a growing. And here these are dendritic cells, so they are not growing. They are differentiated. So what we then looked at is a simpler case of shape change and migration, which is when cells spread. And so you can do that with like this sort of cells that have adhesion molecules. So you put them on the surface, and you can see that the surface increases as they spread on the surface. And what we found with that is that the cells, on average, lose a little bit of volume. Actually, here you can look. These are the spreading area curves, and this is the volume curve of the cells. And the faster they spread, the more volume they lose. Okay. And if you modulate their cytoskeleton to make them spread a bit slower, a bit faster, you can change also how much they will lose volume, and you can really get a significant change of volume in this condition where the cells are non-contractile and they spread very fast. They lose more than 10% of volume. So you can see here that's a quite nice relation between the volume, the speed of volume loss and the speed of area change with this condition, which is a very fast spreading cell that loses a lot of volume. So that's not really explained by Pump-Leak model, because according to Pump-Leak, this shape change should be done at constant volume. But what the model that was proposed actually in collaboration with Jean-François, with Amit, who was a postdoc with Jean-François, and with Pierre Sans for this work, was that actually there is a mechano-osmotic feedback. So there is a coupling between the pumping rate of the ion pumps and the tension of the surface of the cell. So that when the cells are spreading fast, they will change their pump-leak equilibrium. And that could be a priori in any direction, but what we found is that it changes in the direction that the volume is decreasing, okay? Which we propose in this work, that maybe it's a way for the cell to adapt the tension of its membrane when it's spreading too fast, that faster than the actin turnover, so the actin gets tensed, okay? And so it starts to tense the membrane, and the loss of volume would go in the direction to reduce the membrane tension, okay? And we showed that you can actually spread fast without losing any volume if you remove some part of the actin cytoskeleton. So here it's a drug that is affecting the branched actin, or if you drug some ion pumps. Okay, and then you spread, actually you even gain a little bit of volume when you spread in this condition. Okay, so there is this active coupling, let's say, between volume and membrane tension that is responsible for this fluctuation of volume, which is rather small but is significant, and that you could not explain with pump-leak. But if you put this sort of coupling, you can explain it. And now I will show you a last experiment that we did, which was a little bit, let's say, to test, to push the model further, because if all these things depend on ion pumps, okay, so it means it cannot happen so fast. So now what happens if we press the cell really fast, okay, at a millisecond time scale? So here, normally, you would expect that all these pumping questions don't happen, don't have time to take place. So here we go with this 2D confinement. You see from a round cell to a kind of pancake-shaped cell. Here you see the image of the cell. You can see already that the membrane, the intensity on the membrane is going down, so the membrane is expanding. And if you remove the actin cytoskeleton, the cell is making all kinds of bubbles. So when actually you deform it, it doesn't really, you can even already see it on the surface area of the cell. Here the surface area of the cell does not really increase, it's just the size of the bubbles that increase. Okay? And if you measure the volume, you see that in the control condition, the cells are losing volume. Okay? But if you depolymerize actin, they are losing much less volume, except if you compress really a lot. But at this sort of height, for example, here they have lost almost 30% of their volume, while with lateral-inclined A, they have not lost less than 10%. Okay? And so we wondered, so what's going on with these cells? So we recorded, as we were pressing, we recorded as fast as we could the fluorescence around the cell. And what you see is that you have this sort of streams of very transient in a few images, so it lasts a few tens of milliseconds, streams of black thing that is, what is black here is what is inside the cell. So the cells are losing part of their volume as you press on them, just like a sponge, even though it's not clear how these could act as sponges. It's not supposed to really be a sponge. But when you look at the volume change, you really see that the volume is changing from one frame to the other. So it's like the frames here are every 30 milliseconds. And at that point, we thought, okay, maybe it's really a problem with our measure of volume. And so, but when we treat the cells with latranculinase, so we remove the actin cytoskeleton, the volume is perfectly constant. Okay, and the loss is really, this super fast loss is really. So we thought, okay, maybe one solution, easy solution, is that we just lose some cytoplasm, okay, because we make some holes in the membrane. As we change so fast that the membrane gets tense, it makes little holes. The cytoplasm is pressed out, and then the holes close again after. And the cells are perfectly alive. I mean, they don't care at all. You can do that to them. 100% of cells are alive. But if you measure, we have techniques to measure the dry mass of the cell, and the dry mass is really constant. You see, when you press the cell here, it has lost about, here 30% of its volume, but the dry mass did not decrease by 30%. So it's a mystery, I must say. We have been talking about that. I think at the moment we have no solution. So the easiest solution, if you want, is that somehow the cell is losing water and osmolytes, small osmolytes, which dominate the volume, the Van 't Hoff equation. So typically metabolites, ions, metabolites, but somehow they retain the proteins because maybe the hole they make are small enough that the flow, the thing that goes out, this black streak here is mostly the solute, I mean really the small molecules and not the big molecules.
Is there also an effect in, for example, in aquaporins where it could just evacuate water?
Yeah, but it cannot evacuate just water. So this is maybe requires aquaporin, totally. Maybe you have pores that open enough to let the water plus the ions plus the metabolites, okay, like the amino acids, but not the proteins. So that could totally be that. Okay, yeah. It's hard to remove all the aquaporins, but anyway, there are drugs but we have not done that. So it could depend on aquaporin. Then maybe the model would be that you have some sort of holes that are selective enough that they leave, let's say, the water plus the osmolytes out, keeping the proteins. It's quite interesting. For me, it's still an open question, but it's probably something very important because different types of cells are subjected to this sort of super fast deformation when they circulate in the blood, like white blood cells or also circulating tumor cells. They go through capillaries that are very small, and they go through these capillaries at this sort of speed. And what has always struck me is why cells, you can squeeze almost any type of cell. At this speed, they don't die, they don't explode, no problem for them. So maybe thanks to some specific channels like aquaporin that they can release some water plus osmolytes so they can sustain this sort of super fast squeezing.
So here you're using something fluorescent to mark the inside of the cell. And how big is the fluorescent thing that you use?
Outside of the cell? No, we are putting outside of the cell a dextran, a big dextran that doesn't get inside. Yeah. It's inverted contrast.
Could you compute the flux that you would need to relate it to say the number of
aquaporin? Yeah, we could probably to see if it's consistent. I'm not sure it's really well known how much the aquaporins can open and accommodate.
You say they open.
Yeah. We could test that. For these cells, they are known to have a lot of aquaporin. So, and maybe using some drugs, there are drugs that are not very specific, so you could drug a bit kind of all the aquaporins to check if it works. Okay.
But you could also do the opposite, you put something fluorescent inside the cell and you attach it to different sizes of things, and you could see.
To see if they go out, Yeah, to see like having fluorescent metabolites, for example. I think they would go out. Yeah, pretty much clear.
You also attach it to proteins.
Proteins, that's basically that. And we see that proteins they don't go out. That's the dry mass is mostly proteins, so the proteins don't go out. So it's not a
With these experiments, do you know if the density of the cells is changing?
Yeah. So here you have volume divided by dry mass, so it's changing by about 30%. So you lose 30% of volume at constant dry mass. So you concentrate the cell.
Okay, so it has an effect on the mechanical properties.
That I don't know. We've not looked at the mechanical properties at all. What we are starting to look at is whether it induces a bit like you would do in this sort of loss of volume. 30% is what people typically use to induce the formation of condensates. So we are starting to look at that, if this is enough to induce condensate formation, for example. This could be true. When people do hyperosmotic shock. So now we'll switch to the opposite time scale, which is the growth. And so this is really long time, the long time scale. So here you can use the same method to measure the volume of the cell for several days. And then what you would see is that the cells are dividing, growing, dividing again, and you have this sort of curve where you have the volume at birth, growth, division. So that was I was talking about at the beginning, which we call mitotic swelling. So it's a very transient, large increase of volume just during mitosis. Okay? If you do a zoom on that, you see that the cell really, when it gets into mitosis, the volume increases very fast, about 10 to 20%. Then it's sort of flat, and then it loses this extra volume at the end of mitosis. And at the same time, the mass actually stops growing. So the mass goes through a plateau and starts to grow again just when the cells exit mitosis. And that was really our first work on volume, and we found that this was really any sort of cell type we looked at this increase of volume, but it could be really, the range is between 10 to 20%. And some cells are more 20%, some cells more 10. And so we went through different hypotheses, but the preferred one at the moment is the Manning condensation. That I don't know if it comes from you or from Romain, the idea. No. You said you worked on it before, so probably it's from you. But yeah, so Romain explained it all the time to me, but each time I have to remember in which way it goes. But so basically, the idea is that one thing that changes a lot when cells get into mitosis is that you have a lot of chromatin modification that are associated with condensation of the chromosome. But it's not the same thing. The point here that's nice is that it's not the condensation that is important of the chromatin, it's a change of charge. Okay? And this change of charge is mostly a change of acetyl groups that are removed when cells get into mitosis. And so we redid the experiment, that's where we are with experimentally testing this hypothesis. It's just taking cells and putting a drug that is inhibiting the deacetylase. So if you inhibit the deacetylate, you acetylate. And so what we found is that then the nucleus in this case, but the cell probably too, is losing volume by about the same amount, about 10-20%. Which is basically the opposite of mitosis. Okay. So here we think maybe this is the relevance of this particular thing that Jean-François did in his equations for the volume, is a modulation of volume of 10 to 20% when the charges of the chromatin is changed. The reason why you have here 20 versus some 10 is because a lot of these cell lines are cancer cell lines, and they have the double, easily on average double amount of chromosomes, so the effect is doubled. Okay, we have not checked that yet, but there's been some hypothesis why this could also be interesting for mitosis, because maybe this just 10-20% volume change is already putting your cytoplasm in a state that is a bit more diluted, and maybe that's enough to have some effect. We have no idea if this has a function at all. Yeah.
In the curve you showed with the volume throughout mitosis, do you know when the nuclear envelope breaks?
Yeah, it's about concomitant. So here actually you have really this increase is doing a chromosome compaction, but nuclear envelope breakdown happens about the same time. Yeah.
Could this volume change be due to this? Because then you don't have this membrane that selectively prevents things from. You only have one compartment.
Yeah, but there should not be any effect of losing this compartment on the cell volume. And here you have the same change of volume without losing the compartment at all. So I don't know if there is a good way to So for example, there is no easy way to prevent, to have cells that would get into condensing the chromosome at mitosis without opening the nuclear envelope. It's a little bit difficult. But that's why we are more looking at the effect of changing charges in interface cells. And so far it seems to be working, but we have to check. For example, here, what's interesting for that, for the people who work a bit on the nucleus, is that people often assume that if you decondense chromatin, you will increase the nuclear volume. Actually, with the charge effect, it's the opposite. You decrease the nuclear volume. It shows that the chromatin compaction has no importance for the nuclear volume, basically, or the cell volume. It's really the charges. So now I'll get to the main part of it, and I hope I didn't spend too much time on the beginning because, yeah, it's okay. Still have half an hour. Which is really trying to explain these growth curves. So here you have individual cell growth, and you see that it's not really always flat. It's not what I will mostly talk about. So we kind of understand a lot of these fluctuations which are due to the mechano-osmotic coupling. Okay, and that's one cell, let's say. So this, we believe all these fluctuations is mechano-osmotic coupling, and this is a mitotic overshoot potentially due to the exchange of chromatin charges. But what we would like to understand now is like the main part here. Okay, so what drives really this growth of the cell on long time scale, and this, from what Jean-François explained to you before, if you believe his model, what is growing in the cell is that, okay, is these molecules that are accumulating in the cell, and these molecules are mostly amino acids. So we get into this picture that you already saw from Jean-François, that the volume is mostly amino acids, but the mass of the cell is mostly proteins. So how cells can grow without getting diluted or concentrated? How do they grow so that the volume they add by synthesizing amino acids corresponds to the volume they need to keep their proteins in the right density? So the fact that protein density is constant is something that's well admitted in general and has been shown by a few people by different techniques. The density of cells is usually pretty constant. So that's a technique that Jean-François described, I think, in his previous course, where, from the Manalis lab, where they can measure the buoyant mass of cells, change the fluid around the cell to have two different densities, and with that they can get sort of independently, although it's not completely independent, but they have 2 points. So they have mass and volume, and they show that the mass and volume double together when the cell is growing. So the density is constant during the growth of cells. So we redid this sort of experiments. We were using a different technique. So we use occlusion of fluorescence for the volume, and we use this phase camera for the dry mass. And so this phase camera, what they are based on is they produce some interference, and they measure the interference pattern on the imaging plane of the camera. And from that, they get the difference of phase of the light that went through different points on the image. And from that, they can get the sort of optical thickness, so the refractive index times the thickness of the object the light went through. And the refractive index has been shown to be linearly related to the concentration of about anything you could have in cells, sugar, proteins, DNA, salts, with a slope that is pretty much always the same. So if you take the average slope of that, from your phase image, you can get your dry mass. And we know from the composition of the cell that this dry mass is actually mostly the proteins, okay, the total protein content, let's say. And so it's a very nice work from Pierre Bon in the Monterey lab, who did all this work, and they still develop a lot of nice optical applications for this sort of cameras. And so with that, we can get independently the dry mass, the volume. We have also the spreading area, although it's not very informative here. It's more because we know that it can generate some noise. And so you can see that if you average a lot of cells, you have these growth curves. And if you divide the dry mass by the volume, you have actually a pretty much constant density, but with a big sort of change during mitosis, which is due to mitotic swelling, because during mitosis, the mass stops growing while the volume makes this sort of overshoot. So the cell gets diluted in mitosis, and at mitotic exit, they recondense. What is nice is now, because you can show, I didn't show here, but you can show that the growth is exponential if you plot the speed versus the values. So dvdt versus v or dmdt versus m. And so you can look at the rate. So 1 over m dmdt and 1 over v dvdt. And you see that they are nicely, more or less equal during all the cell cycle, except in mitosis where they completely diverge. But when the cells exit mitosis, which is time 0, they are often losing volume at that time, and they are already producing mass. So during this sort of, this is a sort of 1 hour period during which they converge. You have actually an increase of density that often produces a little overshoot, so the cells are often kind of a bit too dense at mitotic exit, and then they take a long time to go back to their density. It's nice because then you can consider really mitosis a sort of perturbation in the density that you can look how cells cope with that. So now the question is really how these two values are related. And there was one idea that came from the PhD of Romain Rollin, which I reveal. I'm sorry because I think you talk about next week. So you will have all the theory next week about that. But the idea is actually very simple. It's just to say that, okay, we know proteins are made of amino acids, but also amino acids are made by proteins. Okay? Especially the one that counts, like glutamate, they are really synthesized by the cell. So you have enzymes that produce these amino acids. And so if you, so with that you can make a model where you have this, your pump leak model, okay? But now your amino acids, they are made by the proteins. So you could think, okay, it's almost the same as saying that the proteins are making the volume. But it's a bit different because basically you can, so that's the first version of putting the two together. That's simplistic. After I will show you that you have to put more to get to the real thing. But you consider that your growth, so lambda m is 1 over m dmdt, okay, is the rate of growth in mass. Is basically an activity of the ribosome times the fraction of ribosome over the total content of protein. Okay, so it's basically fixed by the fraction of ribosomes. And then from this formula, you rewrite this formula, okay, saying that amino acids, which are basically X, are made by some enzymes, okay, which are this EV here. And so this is the same term as, this is basically the dry volume, this is the wet volume, so this is the same term as here. And you make amino acids and you consume them also. And this is, you consider that on short time scale, EV over P, so the fraction of enzymes that make metabolites, or the fraction of ribosome will be constant. And then you have a nice prediction of this model, which is that if now I put that to 0, okay, because I put a drug, I drug my ribosomes, I put that to 0. The model says that your mass growth rate will go to 0, but your volume growth rate should increase. And actually, I will not develop here the thing, but you can quantitatively say by how much it will increase by fitting all from this data here. Okay. So we did that experiment. So if you put your mass growth rate to 0, so here is actually the mass. The mass is constant because here we put cyclohexamide. Okay, we did a dose response to find the right concentration, so our mass is constant. You see that the volume keeps increasing, and after a few hours, it also stops increasing. Okay, but basically you find exactly what the model predicts, which is that you have a jump in the amount of volume increase. So the lambda v, the 1 over v dvdt, is not only staying above 0, it's increasing. And it's increasing exactly by the factor that you can get from your measures, from your growth curves, which is basically doubling. And then that is not in the model, of course. After there is a relaxation, but which takes hours. That is probably due to the fact that these famous, the proteins that make the amino acids are not constant over long time scales. They either degraded or they are inhibited. So this actually looks really like a time scale of protein degradation. So this is, I think it's a quite nice support for the model of Romain, which really says that it's not the same to say that the volume is made directly by the protein or is made by amino acids which are made by proteins, because you can completely decouple the two. And in this context, you get actually a dilution of the cell when you stop protein synthesis. This is just quantification of how much time it takes to stop and how much you increase the volume growth rate. So in the literature on this question, there is actually now some literature on this question. People have more concentrated on some, not on this volume thing, because nobody is kind of measuring the cell volume, but more on the feedbacks on protein production, so on the mass part. So while in the model I showed you, we considered the mass to be basically a sort of constant, the growth rate in mass, many people say that the growth rate in mass is not constant, but is a function of the cytoplasmic concentration. With different type of experiments, these experiments, they are done in extracts, where the people can add different things, so they can change this value here. And what they show is that the protein degradation rate increases when you are more concentrated, while the synthesis rate increases and then decreases, and that gives you a sort of point where you have the stable concentration of your extract. Here is more what Morgan has been proposing, that there is some crowding effect, okay, so that when you get to higher concentration, you will have a decrease in protein production because the ribosomes get crowded. And here is an effect that was proposed by the Scottheim lab, which also showed that there is also an effect of the concentration in the cell on the transcription, not only on the production of protein, but also on the production of RNA. And so you have a lot of reason to think that actually also the mass growth rate should be coupled to the density. So we looked at whether we could see such effect in our experiment by diluting the cells. So instead of stopping the mass growth rate here, we put some water on the cell, and so they swell. Okay? So you have this jump in volume, then an adaptation over a few tens of minutes, and they don't come back exactly to the same volume, so here they are a little bit diluted. Okay? And what you see is that within really a very short time, that's actually here, the time is 1 hour would be 1 here, so in a few minutes, you have your mass growth rate that is more than doubling. Okay? It's multiplied by almost 3. Okay? And then it slowly decreases. So basically here what you have is your density in this experiment at time 0, you swell the cell, so the density drops, okay? And then that's mostly the recovery here of the volume decrease. But what you have is this jump in your mass growth rate as your density is going down, okay? And so we asked what that would give in our model. And so instead of putting that the mass growth rate is a sort of constant, we put that it's a linear relation with the density. So here is dry mass density. Keep that in mind, is the dry mass divided by the volume. And so then you have your mass growth rate, which is no more linear with the density, but has this quadratic curve. And so which then when we fit with the data is really fitting super well our data, even better than before, because we indeed find that we have a 0 volume growth rate for a non-zero density, which here you get here. And here you see that the mass growth rate decreases with density. It doesn't look like super linear. It's a bit more complicated curve here, but so far we have no reason to put any sort of other curve. So we put that in the model. So what is interesting with that, when we got there, is that somehow we find that there are 2 homeostatic modules, one on volume that is basically Romain's model, okay? But also one on density, which might come from any of the other mechanisms that I showed you before, crowding of proteins, not transcription, because the effect, as I showed you here, is extremely fast. It's really a few minutes. So that's something happening at the level of proteins. But what is nice is that together they define a density at which these 2 quantities are equal. So suddenly what we realize is that this, when we combine these 2 things, now we have a target density that comes in the model naturally, which is the density at which you will. So if you do if you have a perturbation, your cells should converge to this particular density, okay, where the two growth rates are equal. So we wondered whether that's the case. So we took the derivative of density as a function of density, and we found, and so here we take the derivative on like a short window, few hours window, okay? And we take the mean density in that window versus the slope for many cells, and we find a very nice negative relation with a 0 here, a target density that's exactly the one that we get here. Okay. So we have the target density in our data, and we also have the model. If you take this value, you can predict how the cells will correct the density across their cell cycle. So that's this curve here, and you see that the data follow quite well. So now it's how much the cell will change their density from birth to division as a function of which density they are born. So basically, mitosis is a sort of perturbation of density, and then they slowly converge to this target density back. And the model is capturing that pretty well. What is quite surprising is that the target density is the same for all cell lines that we have checked. So what it is, we don't know. We also found in some literature that the yeast have the same sort of density. So it could be something that comes from evolution, where this is a sort of optimal density at which the system functions. We have no idea at the moment. One idea I like is the idea that you have really two scales in this system. You have one scale, which is the small metabolites and ions, which diffuse freely and give you the volume with the Van 't Hoff relation. And then you have another scale, which are these big proteins that crowd very easily, and that give you your mass growth rate. So the mass growth rate negative relation with density is given by the fact that the proteins are big and they are very easily crowded, while the small molecules keep diffusing freely, and they are the one giving the other relation. And with the two, you find this target density. It doesn't tell you why you have this number, but it tells you that really with that, you have these two opposite behaviors that give you.
This mass growth rate is kind of a kinetic effect. Is there something like a jamming or so? I mean, does the dynamic become very slow by orders of magnitude? Or
When you
go to higher densities?
So that we have not done. So that we should. The one measure we have not done here is the diffusion of bigger molecules like Morgan does with the gems. So we have not done the gems. So, but the thing is that the gems are basically, in all the papers I've seen, they are basically a readout of protein density. So I suspect we will find that it correlates. Okay, that we will have an increase in density. By which order, I don't know. So I will show you just one other experiment a bit later that shows you that we know it goes through the mTOR pathway. We don't know the mechanism for the mass growth rate modulation. It can be many different things, but what's important is that it goes the other way, and so it's homeostatic also, and it defines its density. Here I have more a sort of analysis that we did actually after that. That's just, this slide is basically to show that we can perturb this by perturbing in different ways. We can introduce perturbations of the growth of the cells by, for example, using rapamycin or things that perturb the mTOR pathway, not to the point that we stop the growth, but we just decrease the capacity of cells to grow fast. Okay? We put the cells at a sort of a slow pace of growth. And if we do that, you see they still grow with this, for example, drugging. Here is a low dose of cyclohexamide that will drug the ribosomes. The cells still grow, but now if we do an osmotic shock, they are not able to jump in mass growth rate. Basically, the mass growth rate remains small. And also, if we do the mass growth rate against density, it's kind of flat. And if we do that, we over a few hours, we completely lose this mass growth rate, this density homeostasis. So now this relation is flat. So the density is no more changing as a function of density. It's kind of flat, okay? And also we lose. So here maybe I go a bit fast, but we lose the effect that when cells are born, there is an increase of CV of density in the first few hours that is normally recovered. But here with this drug, it is lost. So the cells are not able to reduce dispersion in density post-mitotic. And they also don't correct their density anymore across the cell cycle. So I just put this slide together, so it's a bit of a mess, but let's say this experiment tells you that this regulation of density as a function of density, of mass growth rate as a function of density, is really important for homeostasis. Okay. And here is just, I think it's a nice analysis that was done by the colleagues we were working on that. We were theoretician from Milan. I will tell you their names after. They were working also on at the single cell level. So because here I showed you mostly averages, so they looked at what's going on on the single cells. And basically they were asking, do we have really a picture where we have cells that converge towards density, towards the target density, or are they mostly fluctuating around their own density? And so with that you can do two averages. You can do so the average density minus the population average, or the density minus their own average. Okay? And what you see is much smaller here than here, so which means that cells are basically hardly converging towards the target density. Okay? And that's, you can understand because, I don't know if I have I have it here. I don't know if I have it here. No, that's the complete model. But because basically the convergence is a function of the difference between the two growth rates. So as you get close to the target density, you converge slower, more and more slowly. And it's something you can see, I forgot where, here, for example, where only the cells which are far from the target density have a significant convergence to the target density. Okay? And what is nice is that now, basically, how you can complete the model, you can complete the model by adding noise on the growth in mass and volume, and then asking, is this really useful at all with this kind of slow convergence? And what the model tells you is that it's very useful because otherwise your density on long time scale would really diverge. Okay? Because so here you have no coupling with noise, okay, and the noise makes you diverge. Here you have coupling with noise and coupling without noise. So of course, if you have no noise, you get a very nice reduction in that's the standard deviation in the density, okay, and the difference between the density and the target density. So that's all from the model. And if you have the coupling and you have noise, basically what you are doing is you are just able to maintain more or less constant the distribution of density, which is not very narrow, but you maintain the distribution of density. So basically, what it tells is that these two coupling here and this target density is fighting against all the source of noise that you have affecting your density in the system. But it's not really able to make your system go to the target density. Okay? And with this model, you can fit really, with just getting the numbers from basically the distribution of mass and volume, you can fit all these are data and fit from the model, which are, I think it works very well. And so what we believe we have understood with that is that basically, the single cell crack do not converge because the homeostatic process is very slow. Okay, but and before they have time to converge, you have fluctuations like mitosis. But also the track never converge. The process is sufficient to maintain a range of target density, a range of density around the target. So basically, it looks like that. The ones which are far, they converge significantly. Then you have mitosis, you have again a big perturbation, then you reconverge. And overall, you have really what you would call homeostasis, which means that you have a constant distribution of density in your population of growing cells, despite the noise you get from mitosis and from other type of perturbation. I didn't show you here, but we did all kind of other perturbation. The cells are able to compensate. Yeah.
Can you follow the cell for longer time so that you see that they explore the whole distribution?
Yeah, so that we have not done. Yeah, we would like prevent them from getting to a next mitosis to see if they. That's what.
If you follow them through mitosis,
Through several generations.
you would think that the cell
Yeah.
would explode all distribution.
Yeah, so that we have not looked at. Yeah. We have usually a maximum. We have 2 cycles. We don't have so many cycles at the moment. Yeah. So here is this last experiment to show that one thing we were able to see is that when we dilute cells by different ways, we activate the mTOR pathway. So we believe this is how the cells increase their mass growth rate when they get more diluted. And if we perturb the mTOR pathway, we get this flat distribution. So that's just a hint that maybe the mechanism goes through mTOR, although we don't know what it is. And there was this nice paper that I don't know if you will talk about, but where there was this mechanism proposed for a sort of density homeostasis where you have formation of condensates of certain important kinase. Here in this case, it's a kinase which is called WINC1. It's a kinase that regulates ion fluxes. And so here they propose in this paper, I'm not sure how convincing it is, but it's quite a nice idea at least, that when you make an hyperosmotic shock on cells, the cells shrink, you have formation of condensate of this kinase. It changes the kinase activity, it changes the ion fluxes, and then you reswell until you decondense your proteins, basically. And so that protein condensate would be the actual density sensors. And you could imagine the exact same sort of mechanism for mTOR, for example, regulating mass growth rate as a function of density, and that would be able probably explain what we observed. So that's the conclusion. So the cell volume and cell mass are not equal because volume depends mostly on ions and small metabolites, and the mass is proteins. But they are coupled because amino acids are synthesized by protein, and it couples the volume growth rate to the density. And the mass growth rate inversely depends on density. We don't know by which mechanism, maybe condensate of mTOR. I mean, we are starting to work on that with collaborators because we are not specialists of that. What I find really interesting and what we did not expect when we started this work is that together, these 2 couplings, they define a target density. And this target density ensures basically that dry mass density homeostasis in proliferating cells because it fights against different sources of density perturbation that you have in a growing cell, which are in our cultured cells is mostly spreading and mitosis. But you could imagine that there could be others in a tissue, I don't know. And the homeostasis is, I don't know, basically keeping the range of density around this target density. What is this target density? We don't know. Here I just wanted to mention, but as I suspected I will not have time to go through it, I will just maybe just show you the question because it's the other important question for size homeostasis, which is how you couple, how fast you grow and how long you grow. And basically these cells that proliferate, they grow at a certain rate, but they have also cell cycle going on. And so how much they grow from here to here also depends on the time between birth and mitosis. Yeah. And what we and many others have shown in a lot of different organisms is that you have a correlation, sorry, it's not that one, it's maybe the next slide. You have a correlation between this time and the speed of growth. Okay? And basically, the cells that grow more slowly grow for a longer time. Here, what I showed you is the first thing that we measured. That was the work of Clotilde Cadar for his PhD. We measured that had never been done for mammalian cells is to ask what is the relation between the volume that cells have at when they divide versus the volume they have at birth. And we found that many systems that we found, actually, there is a slope of 1 between the two, which defines it as an adder mechanism, meaning the volume in mitosis is the volume at birth plus a certain quantity that is not correlated with the volume at birth. Okay? And this is enough to ensure, again, homeostasis in a population of proliferating cells. But when you see that, you have really, basically, the difficult question, I mean, the question to answer is, we know that the growth is exponential. So smaller cells, they grow slower than bigger cells. Okay? But they add the same volume. So how is it possible? And one of the answers is that they grow for a longer time. Okay? And there is another answer, is that the growth rate is actually also larger for smaller cells. So the growth speed is smaller, but the growth rate is larger. And yeah, I will not detail that. and in this, if you're interested in this paper, it's published already for a long time. So but we also introduced an idea to compare different organisms for how they are able to solve this question by quantifying how much they alter their growth rate as a function of their size versus how much they alter the length of their cycle. And so you can plot that in this. So here basically is a control by time, and here is a control by growth. And you can see that different, basically everybody is an adder, okay, more or less. And some cells are more controlling by growth, some cells are more controlling by time. And even in mammalian cells, you see that mammalian cells, they usually control actually more by growth than by time in the ones we looked at. And so the next question is how this control is ensured. So how cells modulate, couple basically how fast they grow and how long they grow. And here I will not go through that, but the answer we had, I can just show you here, sorry. The one answer that is out there, yeah. Because there is one answer out there, which is, if you want to read about it, it's really put forward by Jan Skotheim, which is basically that you have dilution of an inhibitor of the cell cycle progression. So you will grow until you dilute enough this inhibitor, okay? And that is kind of enough to get this sort of behavior, okay? It's because you have to wait that you have grown enough volume independently of how your size at the beginning. You have a lot of subtleties in there, but and then we were trying to ask whether we agree with that or not. Initially, I thought we disagree because we see also effects of mechanics on the coupling. And so there is one idea we are discussing about the fact that the nucleus could be actually like a balloon that swells, and when it gets tensed, it triggers the next mitosis. But yeah, the very last result we got on that are that actually it seems that in general, whenever the cell is growing more slowly, it's growing for a longer time. And so we designed some experiment with confinement of that where we thought we would disentangle the two. But we had small cells that divide in a short time, but we found out that actually they grow much faster in the conditions where they have a short division time. So at the end, they end the amount of volume. So we still don't know the answer to that. I think it's a very nice question too. And now I will finish. So I realized actually I should have changed it, but that's a picture of the team, but it's a picture of the team with almost nobody of the people who has been working on what I showed you. Except Romain. So at least you come to see the face of Romain, the most famous person of the talk today. So yeah, the other people, so was mostly, I have their names actually, so you can see. So for the density homeostasis, it's really the work of Nishit Srivastava, who is now, he left, he's now working at Altos in UK with Kevin Chalut. So a lot of you know Kevin Chalut, I think. And then all the theory was done by Ludovico, a PhD student, with Marco Costantino Lagomarsino. And we did a lot of all this work with Marco, actually, for years on the adder and all that.
Here's the seminar next week.
Okay, so you will hear, if you come back next week, you will hear Marco giving the theory side. Probably we'll talk about something else because he works normally mostly on bacterial growth. And Romain, of course, has been very important. And then all the thing I talked about at the beginning was mostly the work of Larissa, who was a PhD student and working on all these short time scale volume changes with Amit Singh Vinchen, who was a postdoc with Pierre Saint and Jean-François. And here, I mean, of course, a person who has been working, I cited her on the last part, was Clotilde Cadar, who is now starting her own team at Institut Cochin. And she will keep working on these questions with different systems. She's been really she's very well known in the community of cell size. Actually, at one point she was better known than me. I think I was joking about that. But yeah, she's really very actively working on these questions and very clever and contributing very interesting things. So I think she will keep contributing. And yeah, and thank you for your attention. Hope